Hi everyone, my name is Isabel Houtkamp. I study Bioinformatics and Systems Biology, and I'm currently an intern at the Systems Biology lab at the VU University of Amsterdam. In the next 4 minutes, I'll introduce you to the humanization of the gorilla microbiome in captivity, a research project that is actually set in Artis, at the Royal Zoo located in Amsterdam, and at Microbia, which is the microbe museum that is part of Artis. So we have taken an interest in the gut microbiome composition of the western lowland gorillas that are housed in Artis. And we've done a compositional analysis to identify which bacteria are present in the gut of these gorillas and also at which abundance. So previous studies actually indicated that the captive primate microbiome is very different from the wild primate microbiome. And more specifically, that the captive microbiome seems to converge to a more human one. So here we want to compare our compositional data on the microbiome of zoo-housed gorillas with that of wild gorillas. So we also use data obtained by Gomez in 2015. And here we are looking at the top 10 genera in both microbiomes. And so what is interesting is that really there seems to be a compositional difference between these two microbiomes. So the most abundant genera, these lists, are really very different. And furthermore, we see that lactobacilli really dominate the Artis microbiome, which is not specifically a human thing, but it has been shown before in primates living in captivity. Furthermore, we see also some similar trends when we compare our observations with observations made in studies regarding human industrialization. So we observe a decrease in Osnella and Treponema species and also an increase in Bifidobacterium species. And this trend, this shift, is also found when the microbiome of industrialized humans is compared with that of hunter-gatherer societies. So this is really an interesting parallel, and we believe it is due to a decrease in dietary fiber consumption. So really zoo-housed gorillas, but also industrialized humans, consume way less dietary fiber. So the bigger picture here is that first we want to characterize the primate microbiome. We believe this is important for their health, for conservation, and also to understand evolution. But we also believe that we can contribute to rewilding the primate gut microbiome, also maybe that of humans. And the dietary fiber really is a key component here. So currently we are executing a high fiber diet intervention in Artis. And we are doing a functional analysis here, which means we are looking at the effects of consuming more fiber on the activity of the gorilla gut microbiome. And we also hope to eventually investigate the effects on behavior and take this a step further. Concludingly, we have seen that differences comparing the captive and wild gorilla microbiome mirror effects of human industrialization. And we've also seen the potential of dietary fiber for rewilding purposes. Thank you for your attention.